Today we're doing disorder detectives. This is a way of you doing a karyotype, a simulated karyotype. Okay, let's take a look at the structure of a chromosome. Your lab will look something like this when you first open it up, and your data sheet will ask you to select a chromosome from the cryostorage area, sketch it, and label its P arm, Q arm, and centromere. Let's take a look here at the structure of a chromosome. This chromosome model is pinched together in the middle because both sister chromatids are being held together by a protein disc called a centromere. The upper arms are called P arms and the lower arms are called Q arms. Most of the time, or some of the time actually, the upper arms end up being shorter. So if you can remind yourself, that means petite. The upper arms are P and the lower arms are Q. Now, depending on where the centromere is on the chromosome, that determines these metacentric, submetacentric, or acrocentric chromosomes. So if we take a look back here, if it's right in the middle, meta is a prefix that kind of relates to middle. So this is a metacentric chromosome. The middle here is, the bottom of the chromosome is a little bit lower than the middle, so it's called submetacentric. And the centromere here is toward the top. So acrocentric, almost like head. Okay, so metacentric, submetacentric, acrocentric. And of course, the very tips of your chromosomes are your telomeres, which you will have to label as well. Now, when you get your chromosomes, it will tell you to gently peel them off, and they need to be in a random order. And so the way you order chromosomes, pretty neat that the longest ones go first and the shortest ones go last. So find your longest chromosome, which might be this one, and that should be chromosome 1. And so what you'll do is you'll match up these chromosomes, and you'll kind of see if all of these bands match up. If they don't match up, that could mean a problem. Your data sheet will ask you, are there any abnormalities, such as monosomy, meaning you would have only one chromosome instead of a pair? Do you have a trisomy, a deletion, insertion, translocation, inversion? Let's look at a few examples of what might be wrong. In a trisomy, if you end up with identical chromosomes and there's 3 of them instead of 2, that's a trisomy. If you have an inversion, this is how the chromosome should look right here, but notice looks different. This part of the chromosome actually flipped itself over and is up here, so it's inverted. Another thing that might happen is a deletion. If you'll notice, the chromosome is identical all the way up until you get to here, and then this part of it is missing, so that would be a deletion. Sometimes these are a little more hard to find. A translocation, this chromosome kind of looks like it has a deletion. But then once you finish your whole karyotype, you'll notice that this chromosome is identical all the way down until you get to here and you have this extra band. And so that means that this part of the chromosome was translocated here. And so on your data sheet you would talk about what arm, was it the P arm or the Q arm that had the deletion, etc. Now, you'll also need to talk about what might have gone wrong with this person. You have, this will be what your disorder detective's karyotype looks like when you're finished. You'll look at what case study you have, you'll read about your patient, and you'll put some of this information on your lab sheet. And then based on what you read here and based on your results, you will look at your other sheet that's on the back of your data sheet, and this kind of gives you some hints. Down syndrome is when you have 3 copies of your 21st chromosome. Trisomy 21 is when you have trisomy 18. Edwards syndrome is when you have 3 copies of your 18th chromosome. So you will use that table to try to identify what type of disorder, if any. You may have a normal karyotype that you've been given. Your teacher will probably then have you follow up and research some more information about your disorder. When you are finished, you need to take all of the chromosomes gently, notice it says peel carefully, and put them back in a random order back on your board, and be sure that you have all of them. Check around your workstation to be sure that none of them are on the floor or on your desk. Thank you.